Welcome once again to the Mutation Surveyor webinar series. The focus of this webinar is on quality values. We will discuss what they are, how they can be used in the software, and how to output them. Quality values are used to assess confidence and accuracy of sequencing as well as mutation calling. These scores can be used to help minimize false positive calls by identifying regions of low quality in the trace files. Quality values can be assigned to measure nucleotide quality, such as Phred Score and signal-to-noise ratio values. These scores can then be collectively used to measure the quality of an entire trace. Other scores that help assess the overall trace quality include the base score, lane quality, and signal strength. To evaluate the confidence of a mutation call, scores such as the Mutation Score, Indel quality score, and NM score can be displayed. Now, let's discuss each of these quality values in a bit more detail and see how the software utilizes the scores. One of the most fundamental scores in sequencing is the Phred Score. The Phred quality score is defined as a measure of the quality of the identification of the nucleotide bases generated by automated DNA sequencing. It is logarithmically linked to error probability of the base call, and a score is assigned to each individual base call. The mutation project settings includes preferences for how to handle Phred scores in the project. If loading trace files into the project that have already been processed and base called, the Input tab of the project settings provides options to display either those Phred scores assigned by the sequencing software during base calling, or those calculated by Mutation Surveyor after mobility correction. The latter option is selected by default. The Display tab contains a checkbox that, when selected, will result in the display of the Phred score values below each base call in the chromatograms. These chromatograms with the Phred scores displayed include the sample file view, as well as the sample and reference electropherograms in the graphical analysis display. Mutation Surveyor can also include Phred scores in reports. The graphical reporting options that include the Phred score display are the Project Reviewer report, the GAD report, and the Export GAD report. Several table reports can also include Phred score information. The HGVS report can filter variant calls based on a set Phred score threshold and display trace quality information based on Phred score. When the project includes regions of interest defined in the GenBank file, this report will output additional ROI quality information such as the range of Phred scores and the location and value of the Phred scores that fall below the set threshold. The HGVS subreport mutation output for database includes a setting to display the Phred score of the base call at the position of the mutation call. The custom report is another report option that includes several options for utilizing Phred score to report both nucleotide quality information and overall trace quality. The Filters tab of the report settings provides an option to filter mutation calls at locations where the Phred score is outside of a user-defined range. Settings on the Display tab can be used to look at nucleotide, regional, and overall trace quality. If the GenBank file loaded into the project defines specific regions of interest, settings exist in the Region of Interest section to display Phred score information relative to those regions for each sample. Below the Region of Interest section is the Quality Information section that can provide trace quality information based on a Phred score threshold for selected trace regions. When the average Phred score for the selected region is below the threshold, the report will flag this value for the sample trace file with a red background. The Quality Information section also includes options to display nucleotide quality information for the sample trace files included in the report. After setting your desired Phred score threshold, you can report trace sample metrics based on this value, including the number of nucleotides in the sample with quality values below this threshold, the location of these nucleotides, and a range of Phred scores in the sample. To include the Phred scores of the base with a mutation call, click on the Mutation tab of the report settings and select the checkbox next to Phred score located in the General Information section. The JHU report is a customer report designed to provide quality information regarding regions of interest. The average Phred score for the bases within the designated ROI is displayed as well as the size of that ROI that meets the set Phred score threshold. This report and the custom report are the only two reports that can display the ASCII character associated with the Phred score for each nucleotide of the ROI. The Pretty Base report is another customer report available. With this report, the user can select to include a column which displays the Phred score of the base with the mutation call. Some reports include the option to generate PhD files which contain the Phred score for each called base. These files can be generated from the project report subreports Consensus Sequence Text and Sequence Text Comparison. The Phred score will be displayed in the middle column to the right of the associated base call. The next quality value we will discuss is the signal-to-noise ratio. We define signal as a mutation peak intensity and the noise as the median noise intensity for the minor peaks. The signal-to-noise ratio determines how large the signal must be for a mutation peak relative to the noise for a mutation to be registered. The minimum amount of noise at a given base position is 250 relative fluorescence units. This quality value is factored into the calculation of several scores, including the mutation score, heterozygous Indel score, and lane quality. Since this value does contribute to the mutation score, the mutation table in the graphical analysis display will provide this value under the signal-to-noise ratio column for every substitution call. To report the signal-to-noise ratio of a mutation call using a graphical reporting option, you can use a project reviewer report. The setting to include this information must be selected in the settings. The value will be reported as part of the mutation call located in the mutation table pane of the report. As mentioned, the signal-to-noise ratio can be included in the output of some reports. The HGVS report is one of these reports that provides this output option. In the HGVS report, the signal-to-noise ratio can be used to measure and report trace quality. A threshold can be set to alert users of trace files with average trace quality values that fall below their standards. The output for mutation database subreport also includes settings for reporting signal-to-noise ratio information. However, in this report, the option is to include a signal-to-noise ratio column that provides the signal-to-noise ratio value for that position with the mutation call. The other report to provide signal-to-noise ratio information is the custom report. In this report, signal-to-noise ratio information can be used to measure and report both trace and nucleotide quality. Use the settings available on the Display tab to output the desired information relative to trace information. Under the Quality Information section, use the left settings to output quality information relative to signal-to-noise ratio for the preferred regions of the trace. Like the HGVS report, a threshold can be set to flag any traces with values below this threshold. Use the settings on the right side to output single nucleotide quality information. After setting your preferred threshold, different metrics can be selected to report. The custom report can also output signal-to-noise ratio values pertaining to the mutation calls. The checkboxes for signal-to-noise ratio, located on the top right of the mutation tab, can be used to both display the value for each substitution call and also filter calls based on these values. The base score measures the quality of the comparison region for a sample. The base score displays the average of the Phred scores for the bases within the comparison region and can be included in the output of all standard reports when selected for display using the Output tab of the Mutation Project settings. The lane quality is similar to the base score in that this quality score provides information regarding trace quality. However, the lane quality uses signal-to-noise ratio instead of Phred score and is calculated for the entire trace prior to alignment. Lane quality scores can range from 0 to 100, with 100 being the highest quality. Please keep in mind that a lane quality of 0 does not necessarily indicate a poor quality trace, as this can also be indicative of a heterozygous indel event present in the sample. The lane quality is also used by the software as the deciding factor when selecting between multiple available reference trace files. Let's take a look at the mutation project settings available for the lane quality. Located on the Config tab of the Settings dialog is the Calculate Lane Quality within the Region of Interest setting. Select this setting to have Mutation Surveyor calculate the score relative to the ROI designated in the GenBank file rather than to the entire trace. Two settings are available on the Output tab of the Settings dialog. Selected under Default Settings, the Lane Quality checkbox in the top Output Table Fields section will result in the output of the Lane Quality Score in the standard reports. The column header and the reports for these values is labeled as Quality. In the Quality Threshold section below, leave the Lane Quality Threshold as 0 to not limit output, or set a preferred minimum value for the lane quality. The words low quality is displayed in the quality column in reports for a trace with a lane quality that falls below the quality threshold, and any mutation calls will be hidden for this trace. The lane quality is displayed in the electropherogram pane of the GAD, above each trace and to the right of the trace name. A lane can be deleted by selecting the Delete Lane option from either the Mutation Table context or from the Sample Trace Name context menu. The lane quality for a deleted lane is set to low quality. All of the mutations for the deleted lane are removed from the mutation table. Lane quality values are output in two graphical reports, the Project Reviewer report and the GAD report. Just like in the GAD, the lane quality will be displayed above each trace to the right of the trace file name. Lanes can also be deleted through these reports prior to saving the output. Although not included in the output, the lane quality may be used to help group samples in the clinical report, which is another graphical reporting option. Trace quality is available for second and third order grouping, and when selected, will result in traces sorted within the first order grouping based on the lane quality for the trace. For tabular reports, lane quality will be output in the standard reports when lane quality is selected for display from the output table fields located on the Output tab of the Mutation Project settings, and also in the Custom report when lane quality is selected from the Display tab of the Custom Report Settings dialog. Lane deletion can be performed through these tabular reports through the Context menu option. The project report utilizes lane quality as a filter for some of its subreports, such as the consensus sequence text comparison, and the amino acid text comparison reports. Signal strength is the average relative fluorescence signal intensity for each nucleotide and can be used to evaluate overall trace quality. Output of signal strength is available in the Custom report with the setting located at the bottom of the Display tab of the Custom Reports settings. Please note that Mutation Surveyor does not calculate these values, but instead reports the values that are embedded in the trace file. The last quality scores we will cover provide a level of confidence for a particular mutation call. Confidence is primarily provided through a mutation score. The mutation score is based on the concept of Phred, where the quality scores are logarithmically linked to error probabilities. Mutation scores for point mutations are calculated by the patented anti-correlation algorithm. The values for overlapping factor, dropping factor, and signal-to-noise ratio are used in the calculation of this score. The Indel quality score is the mutation score that is calculated for insertion and deletion events. For heterozygous indels, a heterozygous indel score is calculated using the ratio of signal to noise before and after the start of the event. A homozygous indel score is provided for homozygous indels, and this score evaluates the spacing between the flanking aligned bases as well as signal in the uncalled region. Mutation scores and their associated values are displayed in the graphical analysis display. For two-directional data, the mutation score of the forward directional trace and the mutation score of the reverse directional trace are summed, and the sum score is used to determine if the mutation call is rejected, passed, or put into check. For substitution calls, the mutation score is also displayed in the mutation electropherogram at the location of the mutation peak. In addition to the graphical analysis display, indel quality scores for heterozygous indels can be found displayed at the top of the heterozygous indel detection tool. Graphical reports include the mutation score with the mutation call information, but also display the mutation score in the mutation electropherogram just like in the GAD. This can be seen in the Project Reviewer, the GAD, Export GAD, and clinical reports. Of these 4 reports, Only the project reviewer report can display both the individual score and the total 2D score. Some graphical reports show mutation score information as an average score for a given position. These reports will include an average mutation score pane. In the graphic display report, this pane shows the average mutation score for all of the calls at the indicated position for the contig. In the project display report, the chart shows the average mutation score for all of the calls at the indicated position for all of the samples aligning to the same GenBank file. Individual mutation scores will be reported in standard reports when selected on the Output tab of the Mutation Project settings. The score is displayed as part of the mutation call with a $ symbol preceding the quality value. Mutation scores can also be reported by the custom report. The custom report includes settings to report and filter by both individual and total two-directional scores. The final quality value we will discuss is the NM score. This value indicates the contribution of the normal allele at the position compared to the contribution of the possible mutant alleles at the same position. The score calculation is based on the height ratio of the 4 different dye colors and the shape of the peaks at each nucleotide position. An NM score can range from 0 to 100. Only the custom report and pretty base reports include settings to output the NM score. The custom report can be used to report nucleotide quality information for the trace based on the NM score of each base within the selected region. Just like with the Phred score and signal-to-noise ratio, the custom report can report relative to the NM score for the selected region, how many nucleotides fall below your set threshold, the location of those nucleotides, and their specific score and the range of scores found for that region. Both the custom report and the pretty base report include the option to display the NM score for the mutation call or even for normal alleles in the custom report. A high score, often greater than 5, indicates a high degree of similarity between the sample trace and the reference trace. This in turn means that the sample trace more than likely contains a normal allele at that position. A low score, often less than 0.1, indicates a high degree of difference between the sample trace and the reference trace, which means that the sample trace more than likely contains a homozygous substitution. Scores in the middle, generally from 0.3 to 3, indicate high levels of noise and error or heterozygous substitutions in the sample trace. Now that we have reviewed each quality value individually, let's review the different displays and reports mentioned for each score and summarize what each one can provide. First, we can review what is available in the Mutation Project settings. To open the Mutation Project settings, we go to Process, Settings. The tab on the far left side is the Input tab. This includes settings for the type of Phred score to display. One tab over, the Contig tab, includes settings for reporting lane quality for a region of interest if designated in your GenBank file. The top section of the Output tab includes display settings for your standard reports such as the mutation report. Here we can see the settings for base score, lane quality, and mutation score. Below this section is a filter option based on lane quality. Finally, the setting to display Phred scores is available on the Display tab. The two main displays in Mutation Surveyor are the Sample File View and the Graphical Analysis display. The Sample View can display the Phred scores assigned to the bases when selected for display. The Graphical Analysis display can also include the Phred scores along with the trace's lane quality. In the bottom mutation table, we can find the signal-to-noise ratio for the mutation call and the mutation scores, both individual and total 2D when available. As a reminder, the heterozygous Indel quality score for a called het Indel will also be displayed at the top of the heterozygous Indel detection tool. Next, we will review the quality values available in the graphical reports. Graphical reports can be accessed under the Display menu and some accessed through icons on the toolbar. The Export GAD report takes a screenshot of the graphical analysis display and therefore can output all of the quality values displayed in the GAD. As we just mentioned, this includes the Phred scores displayed in the electropherograms, lane quality of the trace files, and in the mutation table, the mutation score and signal-to-noise ratio values. The mutation score will also be included as part of the mutation call that is reported independently of the captured image. A similar report, the GAD report, can display Phred scores, lane quality, and mutation score when the mutation call information is selected for display. The graphic display report offers an average mutation score pane to report average mutation scores at positions with mutation calls for the selected config. The project display report also provides an average mutation score pane like the graphic display report, but in the project display report, the pane provides average mutation scores at mutation call positions for samples in all configs that align to the same GenBank file. The clinical report can display the mutation score in the mutation electropherogram for substitutions and can help sort the order of the sample output using the lane quality scores calculated for the traces. The final graphical reporting option to discuss is the project reviewer report. The electropherogram pane can display the Phred scores, mutation scores for substitutions, and lane quality. The mutation table pane also has the ability to include the mutation scores, both individual and total 2D, and the signal-to-noise ratio. Finally, let's review the tabular reports and which quality values they output. These can all be found under the Reports menu. We can look at the standard reports first. The mutation report is an example of a standard report. As discussed, the output settings for these reports are located in the mutation project settings. Standard reports can output the lane quality for the trace, as well as a base score and mutation score for mutation call positions. One of the standard reports, the project report, includes sub-reports that utilize and report quality values in a unique manner. The consensus sequence text, the sequence text comparison, and the amino acid text comparison subreports can restrict output based on lane quality. This can be done by entering a non-zero value in the minimum lane quality field. Of these three subreports, both the consensus sequence text and the sequence text comparison enable the creation of PhD files. One file is created for each sample. As we discussed earlier, PhD files contain 3 columns, with the Phred scores displayed in the middle column to the right of the base call. The HGVS report is formatted similarly to the standard reports, but includes different settings for outputting quality values. The main report settings includes a setting to filter variant calls based on a Phred score threshold and can report regional trace quality based on either Phred score or signal-to-noise ratio. The regional quality information can be reported relative to the entire trace, the comparison region, which is the portion of the trace used for variant detection, and any designated region of interest. If no region of interest is set in the GenBank file, and this checkbox is selected, then the software will report quality information for the CDS region. For projects that do include regions of interest, the HGVS report will additionally provide the range of Phred scores within the ROI, as well as the information for the bases that fall below the Phred threshold, including the location and Phred score value. The HGVS report also has a subreport, the mutation output for database. This subreport includes settings to display the mutation score, Phred score, and signal-to-noise ratio for the mutation call positions. Of all the reports available in Mutation Surveyor software, the custom report offers the most number of settings pertaining to quality values. Let's take a look at what is available in the Settings dialog. On the Filters tab is a section for filtering mutation calls based on nucleotide quality, including Phred score. To use this filter, select the quality to filter by and set your preferred range. Any mutation call with the associated values outside of this set range will be filtered from the report. The Display tab includes all of the quality information that can be reported for a trace, which includes regional trace quality information as well as single nucleotide quality information. If your GenBank file defines regions of interest, you can use the Region of Interest section of settings to display quality information specific to this region. These quality value settings include the size of the ROI that meets your Phred Score threshold and the ROI Phred string, which is reported as the ASCII characters. Below this section is the Quality Information section. Located to the left are the trace quality settings. Here, you can enter a threshold that is set relative to either Phred Score or signal-to-noise ratio. Then, set which region or regions you wish to report. The regions available for selection are the entire trace, which encompasses all bases with values calculated after alignment, the comparison region, which includes only those bases evaluated by the software for automatic mutation detection, and the region of interest. If no ROI is defined in the GenBank file and this region is selected, the software will use the coding region. The software will calculate and report the average value for this region. If that average falls below your set threshold, the report will have this cell flagged with a different color. To the right of the trace quality information are the settings for the nucleotide quality information. Again, you can set a value for your quality threshold. This value can be set relative to Phred Score, signal-to-noise ratio, or NM score. Nucleotide quality information can be evaluated for either the comparison region or region of interest. Then, select what information to display. For the selected region, the report can output the number of bases that have associated values below the set threshold, the location and score of those failed bases, and the range of scores, reported as the lowest and then the highest score of the nucleotides. The bottom-most settings in the quality information section are the lane quality, which, as a reminder, is calculated for the entire trace prior to alignment, and signal strength. The mutation tab of the settings includes the settings for reporting quality values specific to mutation calls. The settings at the top allow for display and filtering of certain parts of the mutation call. For quality value purposes, these settings include the display and filter of both individual and total 2D mutation score, as well as a signal-to-noise ratio. Under the General Information section, there are also settings to display the Phred Score and NM score for the mutation calls in any position selected under the Show Non-Mutation Call information. Finally, let's review the available customer reports that can output quality values. Customer reports are specialized reports originally designed as templates for specific customers, but have been made available for all users. Two of these customer reports provide quality value information, the JHU report and the Pretty Base report. The JHU report is designed to work when the GenBank file defines at least one region of interest. The JHU report settings enable the user to set a Phred Score threshold. This threshold is then used to report the size of the region of interest that meets that threshold. This report also outputs quality information for the trace and region of interest. Additionally, this report will provide the ASCII character representation of the Phred Score string for the region of interest. The Pretty Base report, which is the final report we will review, provides different quality value information from the JHU report. The settings of this report include options to output the Phred Score and the NM score for the position of the selection made under the Display section of the settings. This concludes our webinar on the quality values provided within Mutation Surveyor software. For more information or to request a free 35-day trial, please send an email to info@softgenetics.com or visit softgenetics.com. All technical inquiries may be sent via email to our technical support group at tech_support@softgenetics.com. Thank you for watching, and please be sure to view our other available webinars.